Hello everyone. So today I'll be discussing about the production technology of Trichoderma species. So basically, Trichoderma is a biocontrol agent, and it's used in controlling certain soil-borne and seed-borne diseases, and it is effectively used in place of chemical pesticides and fungicides, so as to reduce the environmental health hazards of soil pollution or the chemicals which are degrading the soil fertility. So we have come up with the alternative solutions, and one of the solution is using biocontrol agents, which have effectively proved to be sustainable and quite eco-friendly in eliminating the soil-borne and seed-borne diseases. So in this slide I will be discussing about the production technology of particularly Trichoderma species. About we'll be studying about the nature and mode of action of Trichoderma species. Then we'll be studying about the isolation from the field conditions and its multiplication and enumeration inside the laboratory conditions. And finally, we'll be studying its morphology and also the field application of this biocontrol agent. So, starting the topic. So, what is biocontrol? So, biocontrol, as we all know, biocontrol is the control of certain diseases or certain pests by use of living organisms, and the main aim is to do it in a such a way so that it is sustainable and it is eco-friendly. Okay. Also, in biological control, we use several kinds of microorganisms, be it viruses, bacteria, or even fungi. So all of these microorganisms which prove to be beneficial in eliminating the diseases from the field conditions. So we use all of these in management system, and it also comes under the integrated pest and integrated disease management, which and this is included under the biological control. Okay, and it also reduces the use of chemical herbicides and weedicides or fungicides so as to reduce the hazards. So what is Trichoderma species? If we talk about Trichoderma species, it is a member of the family Hypoceraceae, and Hypoceraceae, it's a member which is having a typical conidial structure, which we'll discuss in the later slides, which makes it very unique, and the classification becomes easy. Also, this comes under ascomycete fungi. Okay, these are opportunistic, which means that they invade the host, they invade the pathogen in a more sustainable way without affecting the host plant. Okay, and they take advantage of the nutrition of the host. So how do they reproduce? So they reproduce by chlamydospores. The chlamydospores are the dormant spores which exist in the soil uh, till the favorable conditions are again there, and they proliferate in mesophilic temperatures of 25 to 35 degree and a wide range of pH. So what is actually the mode of action of Trichoderma? As we study Trichoderma, we study its different biological applications, and we study the its mechanism. How does it act? So while we study these actions, we come to know that it attacks the pathogen in a wide variety of ways. It can also act by parasitism. It can also act by predator prey mechanism. If the organism is large in size, it's called as prey. It's called as predator. So it acts in a way so that it becomes very useful in the controlling of the diseases, and it becomes an agent of bioremediation. So the mechanisms, if we talk about the mechanisms by which Trichoderma antagonize the pathogenic fungi, it is competition, colonization, antibiosis, and direct mycoparasitism. So what is competition? If we talk about competition, like we all know that weeds grow in a field and they compete with the main plant for natural resources like food, like water, like sunlight. In the same way, we use the same property of biocontrol agents so as to control the diseases. Means, when we apply this biocontrol agent in the soil, what does it do? It competes with the pathogen for the resources, and the pathogen starves of those resources, while as this biocontrol agent takes off these natural resources, be it water, be it light, and it leaves pathogen to die of the scarcity of these resources. So this is called as competition, right? It is also called as antagonistic potential. Okay, antagonistic because it acts totally against the functioning of the pathogen. That's what it's called as antagonistic. So it acts as an alternative method against those chemical methods. Okay, so as I talked about nutrient competition, because it competes for food, for natural resources. And what do you mean by antibiosis? Antibiosis means when a biocontrol agent excretes some kinds of chemicals which partially or completely inhibit the functioning of the pathogen. Okay, there are some exudates, there are some toxins, there are some chemicals. Like in agronomy, we use a term called as sorghum effect. I'm just relating it, okay, so that for a better understanding. So if we talk about sorghum effect, what do you mean by sorghum effect? Sorghum effect is when sorghum residues do not decompose easily. Okay, and when we do that, when we when we harvest sorghum, the residues of the sorghum, like the roots or whatever debris is left after the harvesting of the sorghum, they leave some toxins. Okay, and these toxins remain in soil for some period of time, and they affect the cultivation and growth of the succeeding crop. So this is what we call as sorghum effect. That sorghum leaves some toxins in the soil which is detrimental for the succeeding crop. In a similar way, what does this biocontrol agent do? It secretes some chemicals, okay, and these chemicals act in such a way so that they inhibit or they neutralize the action of the pathogen in the surrounding area. That is called as antibiosis. Now, mycoparasitism, like we talk about parasitism. Parasitism means when an organism derives nutrition from the host, okay, and kills the host and survives. In the same way, what is mycoparasitism? So mycoparasitism is the same thing, is same like parasitism, but it's applicable to fungi, which means that a fungi which attacks another fungi. But in our case, in agriculture sense, a beneficial fungus attacks a harmful fungus in the soil. So that is called as mycoparasitism. Okay, so this was mode of action. Now coming to the laboratory techniques of the production technology of this bioagent Trichoderma, what do we use? We use some tools, we use some equipments, so as to enumerate and multiply this Trichoderma, and first of all isolate it. So what are the different materials which we require? First of all, we require laminar airflow. What is laminar airflow? It is a device, it is an equipment which is used to carry out the operations of isolating pathogen, inoculating a pathogen, sterilizing the equipments, all in a sterilized and ambient condition, so that we work in a sterile and micro-free environment. So it's a box-like thing in which there is a UV light. So before operating on the laminar airflow, we just switch on the UV light, and for 15 minutes we keep it as such, and then after 15 minutes we open the lid, and then we start working inside the laminar airflow, so that our media and our inoculum and our specimen does not get contaminated. Okay, uh, so that is the main purpose of laminar airflow. Okay, and we also use different capacities of conical flask, of petri dishes, of cylinders. Okay, like from ranging from 25 ml to 1000 ml for volumetric analysis of the stock solutions. We also use graduated pipettes, we have got 10 to 100 microliter, 100 to 1000 microliter, so for taking small liquids. So these are tools which are required in carrying out the procedure of inoculating our pathogen. Okay, now what are the equipments? As I already said, laminar airflow. Okay, so what is the principle of laminar airflow? We all know that laminar airflow works on the principle of high efficiency particulate air, And what is this? It means that when we work on an laminar airflow, there is a stream of air which comes out. There are different types of laminar airflows, like we have got horizontal and vertical, and the horizontal and vertical laminar airflow are divided on the basis of the positioning of the stream of air. For example, if we work on horizontal laminar airflow, we find that the front facing us, the front of the face of the laminar airflow which faces us, on that front we have got minute pores which are formed, and from those pores the air comes and it sterilizes the air inside the laminar airflow. That's a sterile air. That's why it's called as high efficiency particle air, because it sterilizes the air inside, okay, the environment inside. So that's it. And second equipment is incubator. So what is incubator? It is an equipment which is used for growing the microbial cultures, okay, and it provides favorable conditions of temperature and humidity, most importantly, because pathogen has got a certain range in which it grows. For example, different kinds of microorganisms have got different ranges of temperatures and humidity in which they grow. So particular kind of environment is provided so that the pathogen will grow, and we get to operate on those pathogens, right? So this is incubator. We also call it as biological oxygen incubator, okay. And the next is hot air oven, okay, and it's mainly used for the sterilization of the glassware, and the temperature is 180 degrees, and the materials are kept for 15 to 20 degree Celsius. So make sure while we use the hot air oven, we wear the gloves, because the instruments inside are very hot. So while taking out the instruments, we should wear gloves, and by this we sterilize the we can say the equipments. But what happens is that sometimes we want to make sure that our culture remains completely sterile. For that we do double sterilization. Like, first of all, we sterilize our equipments inside and inside we can say that autoclave under the steam sterilization, and then we can do the hot air sterilization. So in that case we will face double sterilization, and our equipments and our glassware will become double sterilized, so as to carry out the pure contamination free microbial operations. So as I said, autoclave, it is a long pressure cooker like structure, okay, and the temperature inside is fixed, that is 121 degrees Celsius for 15 minutes, and the pressure is 15 Pascal units, 15 psi, okay. So, and water is also maintained, like for example, and it's very crucial to maintain the water inside, because if water level goes down, you cannot sterilize, because it's all about sterilization. The autoclave is a long cylindrical pressure cooker like unit, and inside that we have got a long cylindrical steel bin like structure. So when we take out that bin, we check the water level, and if the water level is okay, we just put the bin again, and that bin has got pores, so that when water gets boiled at 121 degrees Celsius, so the steam comes through the pores and it sterilizes the media. It sterilizes the media, or it sterilizes the glassware, whatever we put inside. Second is hot plate. It's used to mix the mixture of media. Sometimes the media forms clots, okay, while the formation of media we mix several components, and sometimes those clots are formed. So to break those clots and to form a homogeneous mixture, we use the hot plate. Then microscope, everybody knows the function. Now the types of media, this is very basic and crucial. We use different types of media, but in my research work I used few media out of these. But different media used are, first of all is PDA, it's called as potato dextrose agar. Then there is PDB, potato dextrose broth. And the difference between PDA and PDB, potato dextrose agar, is a semi-solid structure, while as PDB it's called as broth, potato dextrose broth, and it's liquid. And there is TSHM, which is called as Trichoderma selective. It's also called as THSM, actually, Trichoderma herzianum selective medium, because it's a media which is particularly used to grow Trichoderma herzianum strain, which is the biocontrol agent which I have worked on. And then there is nutrient agar medium. This can also be used. So these were types of media. Now talking about the brief description about these medias, talking about potato dextrose agar. So for that, it is a kind of trivial method of preparation, and in this we use 200 grams of sliced and unpeeled potatoes, and we boil these slices in 1 liter of distilled water for 30 minutes, and then we add 20 gram of dextrose and 20 gram of agar and mix it, and then finally the media is put in the conical flask and it's plugged with the cotton and wrapped in aluminum. Then it is sterilized. Then the media which we have formed, it is sterilized in autoclave for 15 minutes and under 15 psi 121. So on the table you can see the composition. Like potato infusion we have used 200 gram, dextrose we have used 20 gram, okay, and agar we have used 20 gram, and distilled water we have used 1 liter. So this is how we prepare potato dextrose broth. Okay, now how do we prepare TSM? It's called as Trichoderma selective media. We have got a TSM which is already prepared, okay, in box. It's pink powder like structure, and we dissolve that recipe in 1000 ml of distilled water, okay, and then we add 0.25 gram of chloramphenicol. Chloramphenicol is antibiotic which checks the growth of bacteria, okay, and also we use pentachlorobenzene to avoid the further contamination, and we have to store the media at 45 degrees to prevent the solidification. Okay, so this is how we prepare the Trichoderma herzianum selective media. This is a pink color structure which is formed, and here we have got an important thing. Like I have zoomed in here, inside this utensil you can see these black structures. This is a kind of abnormality that we face while formation of the media, because these clots should not be formed while making of the media. If the clots are formed, it means our media is not prepared well and it's not mixed well. So for this there is a trick, that what we should do is that we should not mix the contents while heating them, but we should first of all mix the content thoroughly, and then we should heat and continuously keep it stirring until the froth formation takes place. So that and when the froth formation takes place, it means the contents are entirely mixed up, okay, and after that we get a clear red color solution which is called as TSM, and we have to keep that for the solidification purpose. So these were the media and the materials. Now talking about the methodology, how do we produce, how do we actually multiply the pathogen? So for this, the first step is sampling and collection. We collect the sample from the field conditions. We usually collect a soil sample from the field, and then we just do the serial dilutions and all that, and then we isolate, and finally we inoculate and enumerate the pathogen. Second step is isolation of the pathogen from the disease sample. Can be a soil sample, it can be any plant part, shoot, root, etc. And then after the isolation, we do the sterilization, surface sterilization technique. In surface sterilization, what do we use? We use sodium hypochlorite solution, and we can also use mercury chloride, but normally we use sodium hypochlorite, okay. So for the surface sterilization, most importantly, and that is 1% of sodium hypochlorite. Then we do the inoculation, and this inoculation is carried under the sterile conditions of laminar airflow, and after the inoculation we do the pure culturing and the mass production. Talking these steps in detail, how do we isolate pathogens from the disease sample? Samples can be from leaves, we can isolate from the soil, we can isolate. So here I have mainly discussed from the leaves and from the soil. Now from the leaves, how can we have to cut this sample into little small pieces, so that each sample has got a portion which contains the disease and the portion which is healthy portion, and then we have to put them in surface sterilant solution so that the whole surface gets wet. Now what do we do actually? We put 2 to 3 petri dishes. In one petri dish we put the distilled water, in other petri dish we put sodium hypochlorite, and then in third we again put the distilled water, so that we can sterilize the plant parts very well. Like first of all we will dip it in the distilled water for some time, then we will dip it in the sodium hypochlorite, and then the distilled water again, and finally they are surface sterilized. Okay, and then they are finally washed, and then they are blotted dry, the samples. And if the sample is soil, okay, if it is soil, then we do the serial dilution. Now in serial dilution we take 5 test tubes. First test tube is filled with 10 ml of distilled water and 1 gram of soil, so its concentration will be 10 power minus 1. So this forms our stock solution, and the rest of the 4 test tubes, in the rest of the 4 test tubes we fill each one with the 9 ml of distilled water, and we take 1 ml from the stock solution and pour it in the successive test tubes. Okay, like in second test tube, then from second we take and drop into third, then third we drop and take into fourth, so as to make the concentrations like 10 power minus 1, then 10 power minus 2, 10 power minus 3, 10 power minus 1. Finally we make 10 power minus 5 concentration, and finally we take sample from the 10 power minus 5, and then we spread that sample on a media and then incubate for a week, and then see the growth of the pathogen. So this is how I have done it. You can see it closely. Here I am performing the serial dilution, and here I have prepared the stocks. This is the serial dilution for isolating Trichoderma species from the soil. Okay, so now comes the concept of sterilization techniques. There are different methods, but usually we follow 2 types of methods. One is wet heat and there is dry heat. Wet heat means steam sterilization, in which we use autoclave, as I already discussed, because steam sterilization is very important. Why? Because steam sterilization kills the microbes, because steam penetrates deeper into the cells of the microorganisms, and kills them from the very micro level as compared to heat. So steam sterilization proves to be the most efficient means of the sterilization. Okay, second is dry heat. As we know, it's called as flaming or baking. Like we use what do we use in it? We use microwave oven, but what happens is that protein hydrolysis doesn't occur in case of dry heat, but it occurs in case of steam sterilization. In case of dry heat, what occurs is that oxidation of the cellular components occur. Okay, so for that, for oxidation we require more heat. So if more heat is required, but the sterilization is not as efficient as steam sterilization. Okay, like you can see an example, like sterilization can normally be achieved in 15 minutes by autoclaving at 120, while as by dry heating it will require 160 degree, and that too for almost for half an hour. So efficient means of sterilization is obviously it is steam sterilization. Also we can use solvents, like I said, in case of surface sterilization we use isopropanol, we use sodium hypochlorite, mercury chloride. So these can also help in the sterilization process normally of the samples. Now talking about radiation also we can use, like I said, in the laminar airflow we switch on the UV rays before 15 minutes. So we can use UV rays, X-rays. These are also the potential sterilization agents which help in the complete removal of the pathogens. Now next comes the inoculation techniques. There are various inoculation techniques that we use, like there is streaking method, there is spread plate method, there is cork borer technique, and there is pour plate method. Streaking method, in case of streaking method, what do we do is that we simply take a media and we take the inoculum on an L-shaped glass rod, and we just make lines or streaks in different directions, horizontal streaks, vertical streaks. we make on the media, semi-solid media, and finally we allow it to grow, and mostly it's used in case of bacteria. Then there is spread plate method. In case of spread plate method, we don't make streaks. What we do is that we just spread it with the help of a L-shaped glass rod. Simply we spread the inoculum, okay. And spread plate is normally used in case of serially diluted samples, because obviously the sample is very dilute and we cannot streak it, so we just spread it. Also we can use pellets. Cork borer technique. Cork borer technique is normally used in case of pure culturing, because when we have our own mother culture, from mother culture we derive a pure culture, okay, subculture. How do we do that? When our mother culture is prepared, we take a cork borer and take small pellets from the mother culture, and out of that we can prepare subcultures or pure cultures. Then there is pour plate method. In case of pour plate method, we pour the media and we mix the inoculum with it, and finally we let it to incubate. So these are all the methods which I discussed. Like, this is streaking technique. As I said, the inoculum is streaked in horizontal and vertical lines. Then I discussed about pour plate method. Like in serially diluted things, we serial dilution. In case of serial dilution, we use pour plate method, and also spread plate method is also used in case of serial dilution. And then there is the final, which I said, by cork borer. We can take a sample which is like this and drop it into the pure culture, into the fresh media, and by this we can pure culture it or subculture it. So this is how I did the inoculation of the media. Okay, here I'm using the graduated pipette from the serially diluted sample. Okay, so what is pure culturing now? Now pure culturing refers to the subculturing actually from the mother culture, because we need different we are actually mass producing, so we will require large amount of inoculum, so therefore we pure culture it. Okay, we subculture it from the, what do we do is that we take an inoculum from the mother culture, and we just make its make its subculture or pure culture. Now there are different techniques of pure culturing. Like, we can use agar plates. Case of agar plates, we use semi-solid media. That's the difference. And we can, on the agar plates, we can use different inoculation techniques. Like, we can use streaking, we can use spread plate. Okay, different techniques, or cork borer techniques on agar plate. Then there is broth culture. In this broth culture, we use the media which is liquid in consistency. Okay, in that we just pour the inoculum. We take the inoculum from the mother culture and drop it in the broth, and finally we get to see the growth after few weeks. And there is a technique which we use. Like, we take this broth in the laminar airflow, and we open the plug and sterilize its mouth, and we keep it in a bit slanting position, and then we just pour the inoculum, or we can just drop the inoculum into, and can just slightly touch it with the surface of the broth, and then again sterilize the mouth and plug it. By this way we get a contamination free culture. So this is it. As you can see, the plate which is in my hand, it is actually a mother culture, and from the mother culture I have prepared these subcultures. You can see, just a second, as I will show you. So this is subculturing. Okay, see, this is the mother culture. This is called as, this is mother culture, and from mother culture I have prepared subcultures. This and this. These are all subcultures. You can see the zoomed picture of the subculture which I have prepared. Okay, from this I have prepared this. Okay, so this is the main original, or you can say mother culture, and this is the subculture. So this is it. And then finally we talk about mass production. In case of mass production, we also have got two methods, like spore suspension method and broth method. So in case of mass production, we consider two things. First of all, there is substrate for growth and there is carrier material, because while you prepare something, we require a substrate with which we can attach our we require a substrate which is an actual active ingredient, and we require a carrier material which actually transports our active ingredient into the soil. So in this case, substrate can be wheat grain, can be sorghum, etc., and carrier material can be talc, vermiculite, molasses. Here we have used talc. Like we can say broth culture method, I have already discussed about it. In case of broth culture method, we just first of all prepared the PDB, okay, potato dextrose broth, which I already discussed, and then we inoculate that broth with the pathogen, and finally we find a fungal mat which is formed on the broth, and then this fungal biomass is collected and it's mixed with the talc powder in the ratio of 1:2. That is, one part of the fungal broth and fungal mat, and two parts of the talc are mixed, and finally this mixture is air dried and it's mixed with the carboxymethyl cellulose. What is the use of carboxymethyl cellulose? The use of CMC is that it enhances the efficiency of the this biocontrol, because when we apply it in the soil, it helps the active ingredient to release very quickly. That's what CMC is used for. So we used it at the rate of 5 gram per kg, and then finally it's packed in polythene bags and should be used within 4 months. So here is the preparation of talc based product. Fungal biomass, as I said already, it's collected and mixed with the talc powder at the ratio of 1:2, and finally it is mixed with CMC at the rate of 5 gram per kg, and finally it's packed and used in within 4 months. Okay, so this is me preparing the talc based product of the Trichoderma. Okay, this is it. So thank you for watching. This was all about the production technology of biocontrol agent Trichoderma. So a resultant discussion shows the and while we just culture this Trichoderma, we also do a weekly inspection of the culture and see if there is any contamination. Like I did the weekly inspection, and I used to find the contamination on the weekly basis. Like this white structure you can see, this is the contamination of Aspergillus. Okay, no, it's not Aspergillus, actually it is actually Penicillium, which I found on this. Okay, and this was the colony growth. So this was the subculture that I already showed you, colony identification, and the color is mostly green, and this was the microscopic structure. So conidia identification, what was the morphological features of the conidia was that the conidia were arranged at the right angles on the conidiophores and had those minute globular structures which we call as conidia on the tips. Okay, the peculiar characteristic of the conidia is that they are sharp, pointed, and at the right angles. Right, if I can zoom it, you can see they are at the right angles, and they are sharp and pointed and are at right angles. So this confirms the Trichoderma hergianum strain as it's observed under 40x light field microscope. So if you can see in a zoomed picture, this is Trichoderma. You can see the cluster of conidia, and you can see the conidia arranged at the right angles on the conidiophores. These were some initial cultures in which a lot of contamination was observed. So this is it. This was all about the production technology of biocontrol agent Trichoderma. Thank you.